Assalamu alaikum everyone. In this video lecture, we will be discussing paper 2 past papers of microscopy from the cell structure, which is a topic of AS level biology 9700. Let's discuss question number 6. Figure 2.1 shows a drawing made from an electron micrograph of two adjacent cells in a leaf. So in the figure 2.1, we can see the drawing of two adjacent plant cells as seen under the electron microscope. Okay, part A. Structure A and B are both visible using the light microscope, but the internal detail of these organelles shown in figure 2.1 is only visible using the electron microscope. So they are saying that the structures A and B that we can see in the drawing, they are also visible using the light microscope, but in the light microscope we do not see very much detail of these structures. So B is mitochondrion and A is the chloroplast, right? Okay, and in this drawing we can see the internal structures of the mitochondrion and chloroplast. We can see a lot of detail because this is a picture taken from the electron microscope, or we can say this is a drawing made from the electron microscope. Okay, so the question is, explain why the internal details of structures A and B are only visible when using the electron microscope and not visible using the light microscope. So as we just discussed that chloroplast and mitochondria, they are also visible under the light microscope, but we see a lot of detail of these organelles only using the electron microscope. So whenever the examiner asks you about the details, so examiner wants you to write about resolution. Is that clear? So as we all know that light microscope has a lower resolution and electron microscope has a higher resolution. So you see more details of the organelles under the electron microscope, and you see less details of an organelle under the light microscope. So what should be the answer over here? We need to mention that electron microscope has a higher resolution as compared to the light microscope. What else do we need to write? Since this question is of 3 marks, we also need to write some other points which support our statement about resolution. So as we mentioned that electron microscope has a higher resolution as compared to the light microscope, what we will do, we will further write that we will write the values of the resolution of both the microscopes. For example, we will say that light microscope has a resolution of 200 nanometers, whereas electron microscope has a resolution of 0.5 nanometers. We will also mention electron microscope has a higher resolution because of the shorter wavelength. Electron microscope has a higher resolution because of shorter wavelength. Because of shorter wavelength, right? And we can also write that because the resolution of electron microscope is high, so you can see the internal membranes in the mitochondria and the chloroplast, right? We know that mitochondria and chloroplast have internal membranes, and internal membranes can only be seen under the electron microscope, right? Because of the high resolution. So we will say that as a result, internal membranes can only be seen using electron microscope, because the internal detail they're talking about is because of the internal membranes. As a result, internal membranes can only be seen using electron microscope, right? Please be careful, you cannot write about magnification in this answer because they are talking about details. Magnification is the number of times you enlarge an object to see the image. We don't write about magnification when the examiner is asking about the details, right? So please be careful, don't write about magnification in this answer. Okay, let's move on to the next question. Question number 8. Figure 1.1 is a labeled diagram of a leaf palisade mesophyll cell as seen with a high quality light microscope. So we can see the palisade mesophyll cell, the drawing of the palisade mesophyll cell as seen using a high quality light microscope. An electron micrograph, which is a picture taken from the electron microscope of the same leaf mesophyll cell at the same magnification, would show more detail than is shown in figure 1.1. So they are asking about the detail, right? Again, we have to mention the resolution. Explain why at the same magnification an electron micrograph is able to provide more detail than a light micrograph. So obviously we will write that electron microscope has a higher resolution as compared to the light microscope because of the shorter wavelength of electron beam, right? We cannot talk about magnification over here because the examiner says explain why at the same magnification. So we need to keep the magnification constant and then we have to explain, right? That why electron micrograph is able to provide more details than a light micrograph. You can also mention that electron microscope has a resolution of 0.5 nanometer, whereas the light microscope has a resolution of 200 nanometers, right? Okay, so this is what we have to write. Okay, part C. The length of the labeled chloroplast in figure 1.1 is 5 micrometer. Okay, so they say that the length of the labeled chloroplast in figure 1.1 is 5 micrometer. So this is the labeled chloroplast and they are saying that the length of this chloroplast is 5 micrometer. Basically this is actual length, not the image length. This is the actual length A. Okay, calculate the magnification of the cell shown in figure 1.1. So we have to state the magnification of the cell that is shown in figure 1.1. So how do we calculate the magnification? Simply we know the actual length of the chloroplast. So what we can do, we can use a scale to measure the length of this chloroplast. This will be the image length when we use a scale to measure the chloroplast. For example, when we measured this chloroplast using a scale, the answer was 0.7 centimeter, or we can say 7 millimeter. So 7 millimeter is the image length. So we will write over here that image length is 7 millimeters and the actual length is 5 micrometer. So what we need to do is that we need to convert the image length into micrometer if you want to calculate the magnification. So magnification is equals to image divided by actual, or we can say image length divided by actual length. Image length is 7 millimeter. We need to convert it into micrometer by multiplying it by 1000 divided by the actual length. So let's calculate the magnification. That is 7000 divided by 5. So what is the answer? The magnification is into 1400 times, or we can say 1400 times or into 1400 is the magnification, right? Okay, so basically this is the magnification of the chloroplast, and obviously if chloroplast in this drawing has a magnification of into 1400, we can say that the cell will have the same magnification. the examiner asked us to calculate the magnification of the cell, right? So don't be confused. The magnification of the chloroplast is same as the magnification of the cell. So we are done with this question. Let's move on to the next. Question number 2. Figure 6.1 is a transmission electron micrograph of a plant parenchyma cell. Okay. So this is a transmission electron micrograph, which means that this is an image taken from the transmission electron microscope. Part A. Explain why the image in figure 6.1 is a transmission electron micrograph and not a scanning electron micrograph. Okay, so why do you think that the image in figure 6.1 is a transmission electron micrograph, which is a picture taken from the transmission electron microscope, and not a scanning electron micrograph, that is a picture taken from the scanning electron microscope? Because if we observe an object under the scanning electron microscope, the image that we get is 3D and we see the surface contours. Always remember that in this the scanning electron microscope shows the surface contours of the specimen, right, or the surface boundaries of the specimen, and the scanning electron micrograph is always 3D. The image that we see in figure 6.1 is a transmission electron micrograph because the image is 2D and we can see the internal structures. So we will write that in figure 6.1 we can see the internal structures of the cell and we do not see any surface contours, right? And we will also mention that figure 6.1, or we can say image in figure 6.1 is a 2D image. It's 2D image and not a 3D image, right? So this is what we write when we are asked to identify that the image that we are observing is a transmission electron micrograph or a scanning electron micrograph. Let's move on to part B. The image shown in figure 6.1 is at higher magnification than can be obtained using a typical light microscope. Okay, so they are mentioning that the image that is shown in figure 6.1 is at a higher magnification that can be normally obtained from a light microscope. So light microscope, as we all know, has lower magnification, and electron microscope has a higher magnification, right? So explain what is meant by the term magnification. Remember that magnification is the number of times an object is enlarged to form an image. Okay, right. Part 2. The actual diameter of the parenchyma cell in figure 6.1 along the line x to y is 35 micrometers. Calculate the magnification of the image. Okay, so actual length is given, that is 35 micrometers, and what do we have to do is that we have to find the image length now, because we have to find the magnification. So for example, when we measured the xy length using the scale to find the image length, it was 7 centimeter, or we can say 70 millimeters, right? So the image length is 70 millimeters as measured by the ruler or the scale, and we have to find the magnification knowing that the actual length is 35 micrometers. Okay, so magnification is equals to image length divided by actual length. Image length is 70 into 1000. Obviously we have to convert 70 millimeters into micrometers divided by 35, so this will make it 2000 times. So magnification is 2000 times, and as we all know that 2000 times magnification can only be achieved using the electron microscope and not the light microscope, because the maximum magnification the light microscope has, or the maximum magnification that can be achieved from a light microscope, is into 1500. Right. So we are done with this question. Okay. So we are done with the major questions of paper 2 from the microscopy, that is the part of the cell structure, from AS Level Biology 9700. Thank you so much for watching. Allah Hafiz everyone. Don't forget to subscribe to my YouTube channel. See you all in the next video.